we get experimental. SciAnts. We're curious, non-judgmental.
Yeah, let's talk about Prairie Voles. Before I tell you about this paper, why are we talking about Prairie Voles? It seems like a little bit of an obscure animal to be studying, right? They did a whole brain map of this animal. It's fair if you're wondering why. We have mice. You have other little rodents that are very well studied. A lot of people, groundwork. If you want to study mouse or a rat, go for it. Why prairie vole? Well, prairie voles have this really interesting and discreet mating and bonding ritual that they do. So unlike a lot of other rodents, prairie voles have this kind of extreme pair bonding that they go through. So once a mated pair is in fact bonded, they will participate in offspring rearing together, but they also do a lot of activities to soothe the other one, whether it be grooming if the other one is anxious, or sharing of resources, and like I said, sharing of responsibilities. It's unusual in the animal kingdom, and they're pretty easy animal to study. All right, so that's why people are interested in the prairie vole. So most of the studies you may have heard about, if it's talking about oxytocin or vasopressin or whatever it is, if it's been done in a non-primate model, there's a good chance prairie voles were involved. Okay, wait, so I did say they have this ritual. We'll talk about it again. The window for bonding is 24 hours, and it's very, very discreet. It's not like in humans where, like, who the heck knows at what point we've bonded sufficiently with, like, it's a whole, drinks and dinners and candlesticks. I don't know, right? Prairie voles, it's real clear. They start mating for the first 6 hours, and it's frequent. They're not bonded yet. That's kind of like the initial courtship. The mating is the initial courtship. That's just part of what they need for the bonding. I don't know what to tell you. After 6 hours, the bonding has initiated. They then kind of go into the phase 2, and by 22 hours, the two are officially pair bonded. And so you can look at, that's a short window of time, and there are these discrete time points where different things are happening. So you can check out what's going on with their brain. Easy to measure. But we have different parts of our brains. Our brains are compartmentalized. It's helpful for a number of reasons. Some of the compartments we call the reward area or the reward circuit. It's the part of your brain that kind of gives you pleasure. Tells you you've done a good job, Like when you eat some fatty food or some chocolate, looking at me over here, chocolate, that can trigger your reward pathway. And you're like, why? So the reward pathways, and when it says you've done something good, it's not like you got an A on your test, although I don't know, maybe. If you're a grade grubber like me, maybe that does activate your reward pathway. But it's like from an evolutionary standpoint, when you do something that is probably good for your fitness, like you've eaten a really fatty kind of food or really nutrient dense for your survival, not in the suburbs kind of survival, right? That's the same brain regions that tend to get hijacked when people are dealing with drug addiction. That's the reward center of the brain is what starts getting changed. Same thing with gambling. All sorts of addictions tend to target the reward centers, but they do lots of things in our day-to-day life also. Okay, when we bond and or have sex, not saying the two are always related, but brain chemistry changes. So the same cocktail of chemicals are being released in both species. The species that bonds for life and the species that doesn't. They both have the same chemicals being released during these activities. However, the species that bonds for life has receptors, so it can receive the signal from these brain juices in its reward pathway. Right? The reward pathway being this stuff that tells us good things are happening. That chocolate was delicious. Weren't those flashing lights at the casino fun? Whatever. It has the things that can receive a signal. It can respond to those brain juices. The species that's not monogamous, that doesn't mate for life, it can receive the signals, it's just not in the rewards pathway. The rewards pathway doesn't get activated because it doesn't have these receptors in it. It can't get the signal from the brain juices there.
So something Chad's pointing out, that it's interesting the chemicals exist in both brains, but only one has the receptor for it.
Well, okay. So they both have receptors for the brain juices, for the chemicals. They're just in different places. So it's not as though the message is being sent out and no part of the brain is responding to it. Even in the species that doesn't bond for life, parts of its brain can receive the signal. It's just not the rewards pathway.
Neil had an interesting question, Lita. Given that there's this rewards pathway that you're talking about, does this mean in any way that voles are somewhat addicted to love?
Oh, interesting. So here's the thing. The rewards pathway activates normally in day-to-day life. It's how we've learned to... Our brain has to be able to tell us when we're doing good things. It's like, here's a star, or here's a cookie, you know. That's our reward pathway. It's just that same pathway because it makes us feel good. The dopamine and the other good things that rewards pathway tells us, it can kind of be hijacked. It can go on overdrive. And that's when we go into the territory of addiction. Most addiction has to do with a change of the rewards pathway. So it's not that anything activating the rewards pathway is an addiction, because lots of normal things, listen, this guy over here might suggest that I'm, jokingly, he might suggest that I'm addicted to chocolate, right? I do love me some good chocolate here. I'm sure my rewards pathway is being activated when I eat a piece of chocolate, but it's not a true addiction in that my rewards pathway isn't kind of being like hyper activated, and which can also lead to sort of a desensitization. But the rewards pathway is being activated. Okay, this is looking at the difference between vole species. Humans are a little bit more complicated, shockingly. But here we go. All right, so what happened is these scientists, just to remind you guys, we have two species of vole. They're very closely related. One is monogamous, like it mates for life. The other one is not. It's very promiscuous. It just goes around breeding with whomever will have it. Okay? They found a lot of similarities in the brain, but what they noticed is the receptors in the rewards pathway was different, just in that little cluster of cells. So the receptors from the monogamous vole, they just slapped those in the non-monogamous, the promiscuous vole species. And they found just by doing that, they could get that vole to act like the monogamous species. They could change a promiscuous, by nature, a promiscuous mole species into one that acts like a monogamous species, just by changing this one little receptor in the rewards pathway.
Scoota's asking about what would the implications of this be, like in the broader term, I guess. Like, how does this apply to humans? And then Agathor has a really cool question, too, of, like, did you do the opposite? Like, did you nerf the receptors from the monogamous one?
I think that's a great question. I think they may have done follow-up on it. I don't recall it being in this particular study where they did that, but it's an excellent question. We always like to see that, to do the opposite to sort of prove the point. The implications. Okay, so it's really hard to say. I mean, the reason we study animals like this is because humans are freaking complicated, all right? We can't do this kind of study on humans. In the government and, ethics review boards around the world seem to get a little feisty about it. And aside from that, we don't have the same clear mating rituals and whatnot that things like these voles have. So how do you determine monogamy for a human being, And the voles, you can kind of control their life experiences up to a certain point. Human beings, so much of our brain chemistry is affected by the environment around us. It's complicated, but...
Sam's wondering, what about voles that are not interested?
If they're not interested at all, not interested in mating, well, I mean, in this case, does this specifically pertain? I don't know. There has been some work on the differences between romantic bonding and like platonic bonding in animals, and there are some similarities in that any kind of social bonding can lead to changes in the brain, but that they do also tend to be quite different from pair bonding versus like other social bonding. Yes, but it seems like on is monogamy and off is promiscuous. Yes. That's what they're measuring because that was their question. Are there a few, so whatever voles, I don't know what number of voles just did not want to participate in mating. But if the voles didn't want to participate in mating, they couldn't be used in this study, because the study was based on the premise of mating.
So that's why it's a simple on-off switch.
Yes. I mean, it would have been interesting if they found that the vole just lost interest. I mean, they do have female at either end and a vole in the center. The vole could have gone over and checked out the new lady, checked out the one that he's mated with before, and been like, not interested, and like, I guess gone to sit in the center of the cage, but they didn't report that happening. And part of that, I think, has to do with selection. I mean, the vole had already mated. So scientists are interfering in evolution experiment. Evolution just experimented on which gene is better for the species, and then it's going to nerf one of those. Well, they have the same genes, they just have them in different parts of the brain. And so they didn't really interfere with the species as a whole. They just took a few select voles of the promiscuous species and made their brain look more similar to the monogamous vole in this one special way, and they found that then their behaviors were similar. But I mean, if you're gonna call that interfering with evolution, that's all scientists do, pretty much. I mean, maybe not behavioral scientists, I don't really know what they do. Like, the rest of us who are like muddling around in like dark caves and not interacting with human beings, pipetting things in the dark with just a flashlight, we're just breaking evolution, I guess. We break biology and then try to figure out how it works. But we're talking about the same species. And so this holds true. This is the kind of experiments that are done on prairie voles. Because they have this uniquely monogamous behavior compared to all of their close relatives. Mice don't have this behavior as well, neither do rats. So it's the prairie vole or bust. And remember, it's also, they have this really discrete window of bonding. We know that 24 hours after mating, they've pair bonded. And there are these other, like, individual steps, predictable times in which different phases of the bonding are happening. So the scientists were like, cool, let's figure out what's going on in their brain. They're just looking at the monogamous guys in this one. And so they do some comparisons to the mouse, but just as a point of reference. So they do, like, some structural studies of the prairie vole's brain generally, because they want to know what is going on with the prairie vole at these different time points. So these are the different time points that they looked at. They looked at time 0, where there was no mating. That's a control. They looked at 2.5 hours where mating has started to occur. They looked at 6 hours, which is one of the key time points. And then they looked at 22 hours when it should have been, bonding should have occurred at this point. And what's interesting as a point of comparison, so they looked at mated pairs, and then to compare that to something else, like a social interaction of some type, the thing they were comparing it to was sibling pairs. So it's still a social interaction. It's not quite mate bonding, but there is sort of a social bonding. And so they're able to try to tease apart what is this unique kind of pair bonding versus just other social interactions. Okay. And this is the big take home. I guess I should first point this out. So unfortunately, you guys, you can't get a prairie vole to sit still for an fMRI, which is what we do for humans. Can't do it. So at each of these time points, they just dissected out some brains and looked for neural activity.
So each of these time points is then a different prairie vole.
Yes. It's 12 pairs of prairie voles at each time point. Dissected out. It would be nice if you could follow an individual through time to see, like, that would be cool, but they couldn't do that. So they dissected and looked for neural activity.
Why can't you just drug them or put them to sleep for the fMRI?
Well, because then they're asleep, and so the brain activity is going to be real different, and who knows how the drugs are changing things. Also, the anesthetics change brain activity.
So the drugs wouldn't just freeze everything during that time point in mating. They would cause a bunch of other changes.
fMRIs look for they need, like, active neural activity. The way they dissected the brain, what they were looking for is actually they were looking for CFOS, which just means there's been recent neural activity. It's a gene. It doesn't matter. It's an immediate early gene for those of you who are keeping score here. But so they're able to do that. Anyway, So they found 68 different brain regions that are activated across this time, which is interesting because in human beings and even in some of the Prairie Vole studies previously, they've only found a handful. So they found 68 discrete brain regions that were activated during this pair bonding. And what is very exciting, brace yourselves, there was no sexual dimorphism between males and females and the activity in their brain. Now, why am I so excited about this? You might be wondering, because if you look into the literature about mate care, like offspring rearing or pair bonding or any kind of chemical release that has to do with reproduction or anything else, everyone's going to tell you, all the literature is going to say, the male brain and the female brain act differently, and that is because they have different hormones governing things. All right, so that's why I was really excited. I mean, some of these brain regions are interesting. They didn't do a lot of detailed follow-up on it. But what I thought was really interesting, like I said, is there's this notion out there, for good reason. Okay, there's this notion out there that men and women's brains are fundamentally different, especially when it comes to things like reproduction, because we have different hormones working primarily, different primary hormones doing things in our brains. But these kinds of studies are harder to do, and I like that a really thorough investigation onto the subject in a species that has really fantastic, clear, discrete events happening, kind of pulled this up. I mean, they found 68 different regions of the brain, and it turns out the male brains and the female brains seem to be working in the same way when it comes to pair bonding in this case. I thought it was interesting. It was surprising if you look into the literature out there, and I thought it was really neat. Okay.
Gouda's wondering, so cross-region impact might explain why diverse behaviors are correlated with the bonding.
Yeah, so we don't know, individual regions of the brain. We don't know. We can make some inferences. Some of the brain regions are not particularly surprising, and some of them are a little bit oh, who knew? We don't know what they're all doing, and we don't know how they're all interacting with each other. But yes, the pair bonding, this mate bonding behavior is really complicated. So this is just scratching the surface. And like I said, unless you're really deep into the neurobiology, this probably isn't going to pique your interest, the details of it. But what I thought, like I said, was really cool is it kind of flies in the face of a lot of conventional wisdom about differences between males and females.